Hi, this is JC and Kirsten, and welcome to our channel, Biochem 101. Today we are going to be telling you everything you need to know about gel-shift helicase assays.
The purpose of gel-shift helicase assay is to identify DNA-protein interaction based on size. Gel-shift helicase assays are important for explaining how DNA can bind to different size proteins or even dimerized proteins.
Properties of gel-shift helicase assays include binding of DNA, hydrolyzing ATP, using energy from ATP hydrolysis for disrupting the hydrogen bond interactions between complementary bases, disrupting the translocation of helicase enzyme along DNA.
ATP-dependent DNA unwinding activity through recombinant helicase supplemented with ATP and partially annealed Y-shaped oligonucleotides. The active DNA helicase separates strands of double-stranded DNA substrate into single-stranded DNA oligonucleotides.
Materials required for a gel-shift helicase assay are listed. In order to perform a gel-shift helicase assay, you must make a preparatory calculations, perform in vitro helicase reactions, perform an SDS page electrophoresis, detect DNA oligonucleotides, observe and analyze bands.
The calculations were done based off the necessary volume of each tube. The calculations are listed in the table.
The next step is to run the in vitro helicase reaction. This is done through combining the reagents based on the previously done calculations. ATP is then added, the tubes are incubated at 37 degrees, and after the stop solution is added.
The SDS page electrophoresis is then run. The samples created in the previous reactions are added to the wells of 8% TVE gel. The staining solution is added after the gel is run in the electrophoresis. It's placed on the rocker and washed with DDH2O. The gel is then placed in the molecular imager to detect the bands.
This is the product from a molecular imager. Problem is, ours never seem to come out right, so we're going to use some basic examples to show the analysis.
This is our representation of a completed gel. The columns are based on each well that was filled. The lightest molecules go to the bottom while the heavier molecules stay on top. Imagine a small mouse running through the gel. Now imagine a large elephant running through the gel. Who is going to get through the gel faster? The mouse, which is why the lighter molecules are on the bottom.
Our first example is very basic. There are 3 wells. The first well is just DNA. The second well is DNA and protein A. The third well is DNA and protein B. The lowest row has to be the DNA band because it is the only thing present in all 3 wells. The second well has another band. This must be protein A, as it is the only other thing other than DNA in that well. But where is protein B? It is shown here that protein B does not bind to DNA.
The second example is a little harder. There are 4 wells this time. The first well has just DNA. The second well has DNA and protein A. The third well has DNA and protein B. The fourth well has DNA, protein A, and protein B. Again, DNA is seen in all 4 wells at the lowest band. The second row from the bottom has a band for protein A. Both the second and fourth well have protein A in them. The fourth well has another band. It cannot be protein B because the third well also has protein B and no band. This means that in order for protein B to bind to DNA, protein A must be present.
The third example is the hardest of them all. There are 5 wells this time. The first well only has protein A and protein B in it. The second well only has DNA in it. The third well has DNA and protein A in it. The fourth well has DNA, protein A, and protein B in it. The fifth well has DNA and protein B in it. The first thing that is seen is that there are no bands in the first well. That is because there is no DNA for the protein to bind to. The second well has just the DNA band and, as usual, is the lightest molecule. The third well has 2 bands, one for DNA and one for protein A. The fourth well has 3 bands, one for DNA, one for protein A, and one for something else. It cannot just be the protein B because the fifth well has protein B but no band in it. This means that, again, protein B is dependent on the presence of protein A to bind to DNA. Thanks for joining us on this week's episode of Biochem 101.
Like, comment, subscribe, and we'll see you next week.